Hello, welcome everybody to the Edexcel Unit 2 lessons. Today I'm going to do this question about microscope. I have selected this question. Let's first read the question. Listeria monocytogenes is a bacterium that contaminates food and causes the disease listeriosis. The photograph shows a colony of these bacteria as seen using an electron microscope. Okay, so what are the things pointed out? Listeria is a bacterium that can contaminate the food, and if this contaminated food consumed by people, they get the disease listeriosis. Bacteria grow as colonies, and we can see the bacteria under electron microscope like this. Okay, you can see it's uh, another thing you can learn here. If I ask you a question, what types of electron microscope used? There are two types: scanning electron microscope, transmission electron microscope. From these two, which type is used to take this picture? Yes, this is scanning electron microscope because you see 3D structure constructed by the electron microscope because it scans the surface and then build up the 3D pictures. Okay, so then the question is the first question: the bacterium labeled X on the photograph is 0.5 micrometer in length. Question number one: calculate the magnification of this image. Let's check again this picture. Okay, you can see a single bacterium named X here, and the length is also indicated here. So we have to find the magnification, right? What we have only the actual size. Actual size is 0.5 micrometer. We always use this equation to calculate actual size, magnification, or image size. But image size, if it is given in the picture, we can measure always the length of the image. What is the equation? Equation is am. I am. So I equal m, right? I equal m. A is actual size. I is image size. M is magnification, right? So image size, magnification, and actual size. Now in this question, we need to calculate magnification. So then we can rearrange this equation: m I divided by a. M equal I divided by a. Okay, then we know actual size 0.5 micrometer. Then we need to find image size. So how we find image size? We use a ruler. Okay, here we have a ruler. Then we look closer to measure it. Okay, here we can measure now. It is 1, 2, 3, 4, 5, 6, 7, 8. 8 millimeter. The length of this bacterium is 8 millimeter. Okay, right now we write here 8 millimeter. Now, the problem is image millimeter, actual size is micrometer. We need to bring the unit into one type of unit. So what is the conversion? I'm always telling you to remember the unit conversion because this is not given in the exam. So what you need to remember, 1 millimeter equal 1000 micrometer. This should be remembered, right? Okay, then if you can remember this one, we can rewrite again 8 convert into micrometer 1000 divided by 0.5 micrometer. On this side is also micrometer. Right, now I use the calculator. I divide 8,000 by 0.5 micrometer. My answer is 16,000. Okay, so this carries only 1 mark, but it is always advisable you show the method, and this is your answer. Because some of the questions uh, you get 2 marks, so 1 mark for this, 1 mark for this. So if you write method correctly and the final answer is wrong, then you get still 1 mark. But in this case, it is 1 mark. Now we proceed to the next part of the question in the next section.